Hello there! In this video, we will be learning about mitosis and meiosis and how they compare to each other. Mitosis has 5 different stages. These include interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Interphase has 3 different checkpoints, G1, S, and G2. First up is G1. In G1, the cell steadily grows, develops, and gathers nutrients. In this video, the blue Play-Doh represents the cell, the yellow is the nucleus, and the beads represent the chromosomes. In the S stage, DNA is synthesized or replicated. In this stage, each chromosome becomes composed of 2 identical DNA double helix molecules. Each double helix is called a chromatid, and the 2 identical chromatids are referred to as sister chromatids. During G2, the cell checks DNA for any damage that might have occurred during replication, and it also ensures that the proteins needed for cell division are present. In prophase, chromatin condenses into chromosomes. Centrosomes then duplicate and migrate away from each other, forming spindle fibers. Finally, the nucleus begins to dissolve. In prometaphase, kinetochore spindle fibers stretch out from each centrosome and connect to the chromosomes. During metaphase, chromosomes line up along the center of the cell on what's called the metaphase plate. In anaphase, sister chromatids are ripped apart and become daughter chromosomes. They are then pulled towards opposite poles by the kinetochore spindle fibers. This is also the shortest phase in mitosis. In telophase, the spindle fibers begin to disappear, the nuclei appear, and the cell begins to contract in the center. The final step in mitosis is cytokinesis. As the cleavage furrow begins to widen, what's called a contractile ring forms in the center, helping the cell pull into 2 different parts. This whole process results in 2 independent daughter cells. Up next is meiosis. Meiosis has 2 stages, stage 1 and stage 2. The main purpose of meiosis is to reduce the chromosome number from 2n to n. Meiosis requires 2 nuclear divisions and produces 4 haploid daughter cells. It also has the ability to create extreme genetic diversity because chromosomes are able to mix up with each other. After going through interphase, cells now enter prophase 1. Unlike mitosis, chromosomes now do something called crossing over, which means that they switch pieces of themselves with other chromosomes. This creates much genetic variation within the cell. Up next is metaphase 1. However, unlike mitosis, the 2 kinetochores of a duplicated chromosome are attached to the same kinetochore spindle fiber. Also, even the way that the chromosomes align themselves along the metaphase plate results in even more genetic diversity for the cell. During anaphase, the homologs of each bivalent separate and move to opposite poles, but sister chromatids do not separate yet. This reduces the chromosome number from 2n to n. In telophase, 2 haploid daughter cells begin to split apart. Please note that they are not identical to the parent cell. Last but not least is interkinesis. This is a sort of interphase for the cell. It rests and prepares itself for the second nuclear division, but DNA replication does not occur here. Following this is meiosis 2, which is nearly identical to mitosis. However, the only differences are that in meiosis 2, the haploid number of duplicated chromosomes align themselves at the metaphase plate, and accordingly, 4 haploid daughter cells which are not genetically identical result. The rest of the process continues as usual. Thank you for watching, everyone, and have a great day.